Okay, in this one, we're going to talk a bit about the splicing of pre-mRNA, so that's just a transcribed gene, splicing it into post-mRNA, which can then be translated at a ribosome. And then we're going to talk about how mutations to a functional gene can result in Duchenne's muscular dystrophy, and I thought I would tag the muscular dystrophy onto this one because one of the causes for muscular dystrophy in terms of mutation can be what we call a splice site mutation, which occurs where there's a mutation to the intron-exon junction, which prevents RNA splicing from taking place, resulting in a dysfunctional gene product. So it kind of puts into context why the splicing is important for the production of functional genes. So, unlike in eukaryotes, prokaryotes lack a nucleus, and transcription and translations occur within the same compartment of the cell, whereas in eukaryotes, transcription takes place in the nucleus and translation occurs at the ribosomes in the cytoplasm, mainly around the rough endoplasmic reticulum. Prokaryotic genomes also lack repetitive elements, which we talked about before, about them not having any tandem repeats, and they also lack introns, which are non-coding regions of DNA within coding regions. So they're non-coding regions within a coding gene. Eukaryotic genes do have these introns. They're disrupted by non-coding regions that are removed via a post-transcriptional process called splicing. So you have transcription, you have the gene within the DNA in the nucleus of a eukaryote. You have a DNA helicase, which splits the double-stranded DNA, breaking the hydrogen bonds, holding the double helix together, so you've got your template strand that can be replicated. And then you have your RNA polymerase, which replicates a complementary strand of pre-mRNA from the gene template of the genome. This pre-mRNA product of the RNA polymerase of the transcription, we need to remove the introns because the introns are non-coding regions, and if we leave them in, we're going to end up with a dysfunctional protein. So how do we get rid of the introns? The introns and exons are recognized at sites creatively called intron-exon junctions, identified by 5' and 3' terminus. The 5' end of an intron always begins with a guanine and a thymine, and the 3' end of the intron always ends with an adenine guanine. We call this the GT-AT rule. So if we look to the image on the right, the rectangles in red represent the exons, which are the coding regions of the coding gene. And then the introns between are the non-coding regions of the coding gene, which we are removing. We can see on the left of the intron, there is a GU on the top part. If you look at the strand that says pre-mRNA, you've got the GU on the left and the AT on the right. That's exactly the GT-AT rule, but because it's RNA, there is a uracil instead of a thymine. These intron-exon junctions are recognized by 5 small nuclear RNAs called SNPs, which is kind of what you can call them if you want to just be simple about it, snRNAs, with 50 proteins that form the spliceosome. The spliceosome recognizes these 5' and 3' ends of the intron and cleaves them. So think of the spliceosome as just a restriction enzyme. It recognizes the 5' TT region and the 3' AT region. So 5' being the left side of the mRNA, 3' being the right side, because we always read DNA 5' to 3'. That's just how we do it. So it recognizes these TT and AT regions of the intron and cuts them. This separates the intron from the rest of the pre-mRNA. Once the intron is cut, it loops around itself, as you can see the arrows pointing down from the pre-mRNA, the thing in the middle, to perform a kind of circular loop structure that we call a lariat, and then this lariat just deteriorates. We don't have to worry about it. And then all that happens, the exons, which are the coding regions, a ligase enzyme comes along and joins them together. Then we have our spliced post-mRNA, which can be picked up by a ribosome and translated to produce a functional protein. Now, with regards to Duchenne's muscular dystrophy, it can be caused by a wide array of mutations, like deletions, where you can have certain regions of the exons that have been deleted, or certain regions of the splice sites that we just talked about, the TT-AT regions, or point mutations. It doesn't really matter. You just need to know that a mutation in the dystrophin gene is going to result in a non-functional dystrophin protein being produced. So mutations in the dystrophin gene, which you can just write DMD gene in an essay, but make sure to clarify that that's what you're talking about before you start using it in shorthand, results in the protein being dysfunctional. Since the protein is responsible for stabilizing muscle fibers, the muscles themselves become weak and more susceptible to damage. The damaged muscles are unable to repair themselves properly, and they get replaced with fat and connective tissue, leading to weakening of the muscles and eventual loss of muscle function, as the damage cycles kind of continue and accumulate, which we can unfortunately see for the poor boy on the in the blue shirt on the image to the right. You can see he's probably late teenage years, mid to late teenage years. He should have a higher muscle content after puberty, but if you look at his arms and shoulders, they don't have the expected amount of muscle because they've been deteriorated and just kind of replaced with connective tissue and fat. So that's why he's in a wheelchair. His muscles are very weak, and they're going to get progressively weaker as damage accumulates and he gets older. And then unfortunately, very few of these sufferers of muscular dystrophy make it past 30. Now, it's an X-linked disease inherited only on the X chromosome, so females are generally not likely to have the disease, as they'll have a healthy version on one of their X chromosomes if their mother is also a carrier. The only way that a woman can suffer from muscular dystrophy is if their mother was a carrier and their father was a sufferer, but even then there's only a 50% chance. But it's very unlikely that they're going to have a father who suffers from muscular dystrophy, as by the time they become reproductively viable, their muscles don't work, so they're not likely to mate. That's all. Thank you.